Welcome to a Rice University digital media production. For more information about us, please visit our website at www.rice.edu.
All right, I want to welcome everybody. Thank you. I'm Rick Wilson. I'm standing in for Susan McIntosh. Susan sends her regrets. First, she's recovering from a brief illness and will feel better. And second, she regrets foisting me on the audience. So bear with me and we'll get going here shortly. It's my pleasure today to introduce Dr. Gordon Mills, Professor of Medicine and Immunology at the University of Texas MD Anderson Cancer Center, where he's also the Weiss Distinguished University Chair in Cancer Medicine, the head of the Kleberg Center for Molecular Markers, and I don't know how he has the time, but he's also the chair of the Department of Systems Biology. Dr. Mills received his MD from the University of Alberta in 1977 and his PhD in biochemistry in 1984, also from the University of Alberta. He left a position at the University of Toronto in 1994 to come to Houston and to come to MD Anderson. He served on numerous steering committees and advisory boards and has received a number of honors and awards in his career. For example, while in Canada, he received the Medical Research Council of Canada's Scientist Award. In 2006, he received the inaugural Wan Ki Hong Award for Mentorship, and in 2009, he received the Wan Ki Hong Award for Excellence in Team Science. As an active researcher, he's currently participating in 9 grants or contracts that I could count. This number has only recently shrunk since in the last 6 months he's just ended an additional 7 grants and contracts. As of yesterday, he's co-authored 344 articles in peer-reviewed journals, and there's another 41 that I counted that have been submitted this year. That was as of yesterday. Dr. Mills indicated he's been working feverishly this morning. Among his recent publications is Gene and Protein Expression Markers of Response to Combined Anti-Angiogenetic and Epidermal Growth Factor Targeted Therapy in Renal Cell Carcinoma, which just appeared in the Annals of Oncology. For those of you who would like to look that up, I think it was in January of 2010. And as well, his publications that I've indicated really don't count his invited articles or his book chapters. Dr. Mills has been an associate editor on numerous journals and is currently senior editor of Molecular Oncology and senior editor of Oncogene. Is that right? Okay. Dr. Mills, by any measure, is not a failure. However, he is here today to talk about failures in pressing forward the way in which we do science. So what I'd like to do is have you welcome Dr. Mills, and I'll turn the podium over to him.
Thank you. It's a pleasure to be here today. Hopefully this won't be a failure of a talk either. Okay, so I was asked to present last August, and as every time you're asked to present 3 or 4 months ahead, you say no big deal, no problem. And then I start to ask myself, what in the world is a Scientia with a theme of failure? I like to talk about successes. We try and be successful. And I literally sat there and said, why in the world was I asked? Was this a comment on my research? Career advice? Where are we going here? Okay, so what I want to talk about today, though, are a few things that really relate to the concept of failure. And I think we have to think very hard about what we do in academia today and why we are failing to move ahead as quickly as we can. And I come from the MD Anderson Cancer Center, and I want to put the point here is that we are a little different. We are clearly a university and a university center, but we also have a covenant with our patients to make progress and make progress quickly, and we are failing. We are nowhere as moving ahead as quickly as we can. And if we think about it, the first statement I would like to make is that we have to fail. And if we're not failing, we are not doing sufficiently high-risk, high-yield research, high-risk, high-yield patient care to make a difference. If we are not failing, we are far too conservative in what we are doing. But we have a problem. Academics do not deal well with failure. Go to the PTC for promotion and tenure and say, I failed, and see how far that will get you. Go to your granting agency and say, I've got a high-risk, high-yield study, and see what you get back from them. We have a problem. We have become far too conservative. We are risk-averse, and this is really restricting our progress. I'm also going to talk about the concept of learning from our successes and failures. And we don't do a very good job of learning from either. Frequently, we get the wrong message, go down the wrong path, and make mistakes. We have to pay much more attention to the lessons that we should be learning. If we look at cancer and cancer care, our current trial approaches look at hundreds and even thousands of patients. And in all cases, you're asking, what does the average do? I don't really care about the average. I want to know about the people that I helped. Can I find Mrs. Brown who looked like Mrs. Green that I cured? I don't want to know about the patient that lived 1 week longer. Yet our trial designs are set up to find 1 week longer, not Mrs. Brown or Mrs. Green that we cured. We have to change our approaches and change our concepts. One of the things that I thought you might be getting at when you asked me to talk was the concept that cancer is a failure of cellular homeostasis. That all the things that make your cells behave the way they should, act as social beings so that one cell works together with another. When a cell becomes sick or isn't working right, it dies and gets out of the system. Cancer cells forget that. Cancer cells are selfish. They are not social. They develop their own environment. That whole approach is the concept of systems biology. And as I put the talk together, I said, yeah, that's kind of boring. We're going to talk about other things. It's math and math modeling, and that is really challenging in an audience. I'm also going to talk about what the MD Anderson Cancer Center is doing in terms of a big, high-risk project. We actually call it Project T9, and on about every second day we call it Project Insanity, and I'll tell you a little bit about it later. And I'm also going to talk at the end about one of the big challenges. We have come to a society where we are looked at everything we do. We have compliance issues with being audited for our financial aspects in our grants, making sure we're doing exactly and only what we said, where we're being audited by IRBs, where we're being audited by CLIA and other approaches. And these approaches were developed for 10 and 20 years ago and are not keeping pace with the change in where we are today, and they are causing us to fail in what we need to accomplish. And if we think about this, if we're going to move forward, we cannot be looking at incremental steps. We need to dream no small dreams, for they have no power to move us forward or move the hearts of men. We really need to think big, take risks, and make a difference. Now, Sam Beckett, I think, said this best. If you've ever tried, you've ever failed, no matter. Try again, learn from that failure, fail again, and fail better. And actually, I think this is one of the mottos that I've had for a long time. I've had the luxury and the position that I have at MD Anderson to fail. Now, we have to succeed occasionally too, but that success has been high risk, high yield, and I've been able to take those risks because of the environment that we have and that I've been in. And I hope that many of the people that work with us and that we are training are also going to go out and do those types of high-risk studies that are going to make a difference. And then there's the NIH study section. I have received how many grants back that say it's overly ambitious and there is no guarantee of success? And I seem to remember being ambitious is good, so being overly ambitious is what I want to be. I don't want to be boring. We have to figure out how to change that culture. And if there's a guarantee of success, it's boring. There's no question. It's incremental. It's not something new. It's not risky. And it's not going to make a difference. So we have a cultural problem at almost every level that we're going to have to change if we are going to fail and by failing, succeed better. Okay, so where are we? I'm going to talk today about personalized medicine, the challenges, the opportunities, and how we're going to move this forward. And basically, we are changing the way in which we are thinking about how we manage patients. I'm going to talk primarily about cancer, although in the introduction will be a little more broadly. So is this the right drug for you? Is this the right drug for you today? Is this the first time, the right treatment? Are we getting to where we need to go? Over the last 30 years, we've treated patients pretty well the same way. If Mrs. Green comes to me with an ovarian cancer, she will receive cisplatin and Taxol. We know that 25% of the time she will have no benefit from that at all. As a matter of fact, it will simply be a toxic drug that will make her sick and less likely to be able to tolerate the right drug the next time. So we have to stop treating everybody as an average, that same question I talked about in clinical trials, and start to understand why some people benefit and some people don't, so that we can look at each patient and each tumor and say, is this the right treatment at the right time, and get it right the first time. Now, how are we going to get there? Well, this isn't new. This is a fellow Canadian. In 1892, Sir William Osler made the same comment I think that we're making today. If it wasn't for the great variability among individuals, medicine might as well be a science and not an art. One of the challenges that we have is we are generating an incredible amount of data. It's not information, it's not knowledge, and it certainly is not wisdom, but it's an incredible fire hose of data coming at us that we are going to have to learn to deal with if we are going to be able to benefit our patients. And it's always going to be the physician and the human brain that synthesizes that data into something useful. Now, where are we today? We treat many different diseases. And if we look at the major diseases that we handle today, hypertension, heart failure, and look at the drugs that we use and ask how many times are patients benefiting and having no activity, it turns out that we really are not doing much with many of these drugs. If you look at asthma, only 30 to 40% of patients, in some cases, are benefiting from the drugs that we give them. We need to change the way in which we do this. And if we just simply look at the excess money that is spent on these group of patients that we're not helping at all, and I apologize, I don't think that's showing, oh, here it is showing on this one, that are not being helped at all, it's an incredible waste. These are billions of dollars in each of these cases where patients are paying for and receiving drugs that are not helping them. It turns out that this is not related to these big diseases. In a survey done in 2006, and estimates of the cancer drugs that we use today, both targeted therapies and chemotherapies, somewhere between 60 and 70% of patients in many of these cases are not benefiting from the drug at all. It's the best we've got. It might help some patients. It certainly does something to the average, or it wouldn't have been approved. But we've got an incredible need to do a better job to get the right drug to the right patient. We have another problem. It's a problem in a couple of ways. Some of the drugs that we use today are toxic. Now, some of the drugs are toxic to an incredibly small portion of patients. If you look at this group down at the bottom, these are drugs that were removed from the market where less than 1 in 1,000 patients had problems with this particular drug. Highly active for 999 of those patients, but because we couldn't identify in advance the one patient, the Mr. Brown, who wasn't going to do very well on that drug, we took the drug off the market. Again, being risk adverse, but mainly because we weren't matching the right drug to the right patient at the right time. So where are we moving in terms of cancer? Well, we have two problems with cancer that are major. First, it is a heterogeneous disease. It's polyclonal. It's polyclonal between individuals. It's heterogeneous between individuals. And further, within each tumor, there are probably 10 to 20 separate tumors that we have to deal with that have different genetic abnormalities, different integration of information that we're going to have to deal with. Indeed, that's probably why we're having such difficulty in curing cancer. If we think about it, Mrs. Green's cancer truly is different from Mrs. Brown's cancer, And we may have to find something to treat each one of those independently, or at least come down to smaller and smaller groups that are more likely to benefit from the drugs that we're using. But if we also think about that conceptually, any patient population is composed of a set of people who are responders with adverse events. We clearly don't want to give them that drug. We want to give them something else. Another group who doesn't benefit from the drug at all. And then the worst group of all, of course, the tumor you don't benefit from the drug in terms of the tumor, and it's also toxic to the host. So if we look at this group, the only group we should be treating with this drug is this 1 out of 4 over here. In some cases, it's 1 out of 100. And how are we going to identify those individuals to get the right drugs to them in an efficient manner? If we think about it, many of the approaches that we use today by testing large populations of patients and only a few benefit also deny us from finding drugs that work at all. We have to, if we're going to move forward, identify patients in advance who are likely to benefit and only put those patients in our clinical trial so we have a chance of seeing that we have efficacy. It'll decrease costs, it'll improve outcomes, and greatly increase the speed at which we get effective drugs to the right patients. Yet we're not doing this yet. Why are we failing? Well, I want to go back to this statement. There is an incredible biological complexity and heterogeneity in cancer. That means that we have to develop technologies that look at the tumor that Mrs. Brown has and her own genetic makeup and puts those together and says this is the right approach. There are hundreds of aberrations in each cell. How are we ever going to understand the complexity that that generates? And then how are we going to determine the interactions between the tumor, the environment, and all of the other things that are going on to move forward? Strikingly, we have some great examples if we pay attention. Okay, so cancer is a disease of genes. We need to let the patient teach us what is important, and we have to pay attention to what the patient is telling us. Ron DePinho said a few years ago, and I don't think he had any idea, we have to beware the tsunami of information or data that is about to hit us. We can today characterize your complete genome, every single one of the 3.6 billion nucleotides in your genome, and independently, the 3.6 billion nucleotides in your tumor and all of their changes for now in the range of $3,000 to $4,000 at a time, and do it in 7 days. How are we going to deal with the incredible amount of data that we have on why you are you, why you are you, why your tumor is different, and what are we going to do with this? And this is just talking about the DNA level. So at the DNA level, we know that there are many different aberrations. We're looking at 20,000 genes. That's a number we can think about, certainly far less than my mortgage. We look at RNA. It gets a little more complicated. There's probably about 200,000 forms of RNA. We have great technologies to look at DNA and RNA today. Protein, though, is the actor. It's what makes things happen in the cell, and we really do not have good technologies to move forward. One of the things that we do in our laboratory is attempt to develop technologies to move that forward, and I will not talk about it today other than very briefly here. And this is one of the most exciting events and one of the most challenging events that's happening right now. This is the Cancer Genome Atlas Project. The Cancer Genome Atlas Project out of the National Cancer Institute will characterize 500 cancers and 500 tumors in each of 22 different cancers at a depth and breadth that we've never seen before. We will look at the complete genome, the open reading frames, methylation, copy number changes, splice variants, and our lab at the MD Anderson Cancer Center will do the proteomics analysis and all of the tumors that are coming through the system to integrate at the DNA, RNA, and protein level to give us an atlas of what can go wrong in a patient's tumor and what can happen and what we need to understand if we're going to deliver on the promise of personalized medicine, if we're not going to fail. We have another unique opportunity that's happening today. Industry has become highly efficient at developing drugs. We have for the first time a blueprint of what's going on in a cancer cell. And at the same time, there are over 800 medicines or drugs and vaccines that are either in clinical trial or about to enter clinical trials. And the majority of these are aimed at targeting the specific abnormalities we can pick up by sequencing. So we have this opportunity of putting the two together and making a difference. We have the opportunity of moving from the blunt technologies we've had of chemotherapy and radiation therapy through this perfect storm of new technologies that will let us look at the tumor and the drug toolkit that is available to make a difference for our patients. One of the things that we've learned over the last 20 years is that cancer cells are not particularly robust. Through phylogeny and through development, all of the genes in your body have been set up to work and work efficiently. Those genes have been selected to be in the appropriate homeostatic level, to have the appropriate controls. And when those are lost through mutation and aberrations that happen in cancer cells, both to drive the cancer cell and also because of the underlying genomic instability in the cancer cell, those cancer cells actually are not as robust. And if we can understand why they're cancer cells and what's driving them and gum up that works, we have the potential to target the cancer cell and make a difference without excessive toxicity to normal cells, moving away from killing everything that grows rapidly to hopefully targeting what's causing a cancer cell to exist. And I want to tell you a couple of stories of where we are today. Some successes, some failing to learn from successes, some blatant failures, and then some of the things I hope we're learning as we go forward. And this is one of the most exciting stories that's come out in the last 20 years. In 1962, Janet Rowley, looking at chromosomes in cancer cells, chronic myelogenous leukemia, picked up an odd-looking chromosome. It was called the Philadelphia chromosome. Curiously, she was working in Philadelphia at the time. It was a translocation that brought two genes together. And remember, the chromosome was seen in 1962. In 1986, David Baltimore showed that a little bit of chromosome 22 and a little bit of chromosome 9 were exchanged consistently in chronic myelogenous leukemia. Over 95% of chronic myelogenous leukemias have these defects. Indeed, it's a molecular marker of chronic myelogenous leukemia. We define the disease by the marker, not as much by its behavior. What was exciting is several years later, a drug was developed. It had a number of names, STI-471, Gleevec, and now imatinib mesolit. By the way, every drug that makes it to the clinic has 3 different names to confuse everyone, so we don't know what the history is. But this is what was happening in chronic myelogenous leukemia. This is incremental progress. Back in the 1965 to 1974 era, it was a universally fatal disease. We made progress in 10 years increments. Got a little better, a little better, a little better. We made progress, not as much as we would like to see. And then the drug hit the market. And this is what we're at today. Instead of universal fatality, 93% of the patients will survive their disease, just simply by understanding what was happening and finding the drug to move forward. This really is even more striking when we actually look at not just clinical trial data, but overall data. This is chronic lymphocytic leukemia. We don't have a similar drug. And this is chronic myelogenous leukemia, going along, getting a little better as I showed you, and the death rate just plummets. This is an effective drug. This is what we want to see with an effective drug. So the lesson we thought we had learned is that if you have a target and you have a drug, you have a cure. Unfortunately, we made some mistakes. The first thing we forgot is that the trials that we're looking at is based on the average patient. And in chronic myelogenous leukemia, the average patient had the genetic lesion we were targeting. As we moved to all of the other types of cancers we were looking at, we forgot this. We said, great, now we got lung cancer. We've got a drug that targets the epidermal growth factor receptor. Some lung cancers have the epidermal growth factor receptor expressed, and we're going to have the same success. This is what happened. We actually didn't think very hard because we knew that the fusion gene in chronic myelogenous leukemia was abnormal. The epidermal growth factor receptor in lung cancer was completely normal. It was just present. We also forgot about heterogeneity. So we weren't learning the right lesson from our success. And this is a study with 2,000 patients headed by Roy Herbst at the MD Anderson Cancer Center, major study called Tribute in lung cancer. And these are the patients that were treated, and these are the patients that weren't with the targeted therapy. Absolutely no hint of activity. The drug was not approved, and it essentially lost the market. A few years later, we went back and thought about this a little harder and said, you know what, maybe what's going on here is that there's a population of patients, a subpopulation, Mrs. Brown and Mrs. Green, that benefited from the drug. And we couldn't see that because there was a population that didn't benefit and maybe was even harmed. And we actually know that's the case. This is a reanalysis of that data, showing that if you happen to have not just expression of the epidermal growth factor receptor, but one that's abnormal, just like the BCR-ABL chromosome in CML, you would respond. And indeed, these patients do extremely well for lung cancer. Turns out if you have another mutation, one in a gene called RAS, the same drug, targeting the epidermal growth factor receptor, these patients do worse. Somehow the epidermal growth factor receptor makes the RAS mutant tumors behave better. If you put that together, low number of patients with this aberration, fairly high number of patients with this one, the two together add out and cancel, and we missed an incredibly active drug. And what's even worse, we harmed a bunch of patients because we weren't paying attention. So we failed to learn our lesson. We have learned the lesson a little better, and I want to show you one of the more exciting events that's happened and lead you through the story. So this failure demanded a completely new paradigm. The exciting observation was a new fusion gene, very similar to the one in chronic myelogenous leukemia was found. It was a fusion of two genes, EML4 and ELK, and ELK happens to be a kinase, something we know how to make drugs to. And this is the fusion. You take part of one chromosome, another chromosome, you put them together, and you've got this aberration. Highly reminiscent of the Philadelphia chromosome. Did the same principle apply? And I want to point out that the first time that this aberration was seen was in 2007. We then set out to say, can we develop an assay to find these patients? And we were disappointed. Unlike chronic myelogenous leukemia, where the Philadelphia chromosome is present in 95% of the patients, somewhere between 4 and 5% of lung cancer patients have this particular aberration. A number of assays were developed, and Pfizer, who happened to have a drug in that toolkit sitting there that targeted ELK, wasn't developed for this, but was developed because ELK was important in something else, said if I can find the patients, the right patients, I can make a difference. I can help the patients, I'll get my drug approved. 4% of patients with non-small cell lung cancer have the aberration. They paid to screen 2,000 patients. It costs about $1,000 to screen for this particular aberration in patients today. It was a $2 million upfront investment to find 28 patients with the aberration. I'm going to show you a waterfall plot here today, right now. And what this is a way of doing a continuous analysis of what is going on in patients. And I want to point out that EML4 ELK lung cancers had no response to any known therapy in over 30 years of clinical trials. None. It didn't respond to the EGF receptor inhibitors. It didn't respond to chemotherapy. This is a nasty beast. If we look at what's happening here, this says the tumor grew, and this says the tumor got smaller. For a number of reasons, we use a decrease of 30% as a RECIST criteria to say that we've had an effect. Now, that's only defined because 30 years ago, when RECIST came into vogue, you used the human hand to measure things, and you could pick up a 25% difference. With imaging, we can now accurately pick up a 3 and 4% change, but history rules. We're still using the same criteria, right or wrong. But this is unbelievable. We now have, even with this fairly stringent cutoff, patients with marked responses in a disease with no previous response. By matching the aberration and the drug together, using this incredible new toolkit and the new technologies to move forward. This patient has had the disease go away completely. No hint of disease. This is incredible. 3 years from discovery to making a difference. The FDA, I'm going to talk about them a few times today, that regulate drugs, said this is a non-randomized trial. Therefore, we don't believe it. You have to go out and treat a bunch of patients with the aberration with nothing and a bunch with your drug. And I sit here and I look at this and I say, no patient has ever responded. What am I missing? This should be approved from this. We're going back to old data and old approaches and not learning, because that's the way we've always done it. We need a new paradigm. There's no question that we really are in trouble. This is where we are in terms of the number of drugs approved since 1996 and the amount of money that has been being spent on drug development. Going up at an incredible rate. Since 1996, we've gone from $16.9 billion to $43 billion a year just in drug development. And we've done an incredible job. We've gone from 62 approved drugs to 29. It's gotten worse. As drugs move into clinical trials, we're doing worse. This is the way in which drugs are developed today. An average of 15 to 20 years from when you start to when you actually determine whether it works or not. Not that 3 years that I told you about that should be the new paradigm. And we look at what happens. $1.7 billion goes into any useful drug today. That means you have to have a major market to recover your investment. It also suggests that drug companies in general aren't going to look for just the few patients who will respond. They want to try and treat everyone. Frightening. Once you get into a clinical trial, somewhere between 8 and 16% of the drugs that make trials will pass through the clinical utility. 12 years ago, it was 20 to 25%. We're not learning, we're getting worse. Why are we failing? What are we doing wrong? We need a new paradigm. We have to change the way in which we think. We have to go from this trial design that looks for the average responder, that wonderful lung trial I showed you that was a complete flop, where we look at a broad patient population. It takes 10 to 12 years just to do the trials. We spend $1 billion to do the trial. Success rate of 5 to 10%. Patients that are treated that don't benefit, thousands in each case. Not a very good value. But if we think about these marker-based trials, where we put the patients most likely to benefit and only those patients on the trial, we're going to need fewer patients. It'll take less time to accumulate those patients. It'll cost less money. It'll happen more quickly, and our success rate should go up. And I think that crizotinib example I showed you is the perfect one of how we should move forward. I want to show you some of our successes and failures at the MD Anderson Cancer Center. And one of the key things that we're going to be able to do in the future is let the patient teach us what is important. Not the most recent paper in Cancer Cell, not the mouse model. We're actually going to be able to go in and look at people. We're going to ask why do a very small proportion of patients in early clinical trials of drugs respond. It's 5 to 10%. If I can figure out why Mrs. Green did well on this drug, maybe I can identify Mrs. Brown who's over there and get the same drug to her and make a difference, instead of going ahead blindly, one step at a time, trying to figure out what happens. Well, we thought we had this made. There was new information that came out a little while ago that melanoma, which is increasing in frequency, particularly here in the Sun Belt, has a frequent mutation in a gene called BRAF. It's an aberrant gene. Same sort of story as the crizotinib one, the Philadelphia chromosome. If we had a drug, maybe we could make a difference. So we did a trial of a drug that was developed by a company called Onyx. And that drug was sorafenib, and it had been developed to inhibit BRAF. Long before the mutation was known to exist, somebody had decided to make the drug. We did the trial. We weren't testing for RAF at the time because we simply didn't have the technology, and one patient responded. As a matter of fact, that tumor went away in the one patient. Can we learn from that patient? So here's the response. This is a melanoma. This is the tumor in the lung. And once you have a melanoma in the lung, your outcome is invariably bad. Treated, 2 months of therapy. Oral drug, patient went home, no toxicity, and the tumor was gone. This is a home run. We've got another Gleevec. So we had a targeted drug, we had the genomic aberration, the patient benefited, and we were just touting our success. We knew how to get all the rest of the patients. So we decided to confirm, and we sequenced RAF. No mutation. Oops. Maybe we don't have much of a hit here. So we failed. We failed badly. We now know that sorafenib is a horrible inhibitor of RAF. But can we learn? That patient responded for some reason. It didn't go away. Those tumors don't disappear by chance. Somehow, something in that tumor responded to sorafenib. If we can just figure that out, can we find more patients who would benefit? So we started in. We can now sequence everything that moves, and we sequenced all known targets for sorafenib. It's a fairly dirty drug. Nothing. Started to get a little bit depressed. Sorafenib inhibits kinases. There are about 900 or so of them. So we started sequencing all kinases from the top down, and we found a mutation in a kinase called KIT. It turns out that in certain types of melanoma, up to 70% of them have mutations in KIT. We now sequence KIT for all of our high-risk melanomas at the MD Anderson Cancer Center based on this N of 1 experience, the chance that we paid attention to what the patient was teaching us, and learned from the fact that we failed badly. It was not what we were looking for. Since KIT was important, we decided that we'd better look at KIT broadly. And through the Kleberg Center for Molecular Markers, we have the ability to look at lots of patients, lots of cell lines, and we sequenced KIT in every one of those that we could think of, because we wanted to know the spectrum of mutations that could happen in KIT to determine whether some of them might respond to this drug, that drug, sorafenib, or some new drug. And as we did that, we found a mutation we'd never seen before. And we characterized that mutation extensively, and this just happens to be the molecular modeling approach. The standard drug for treating KIT is imatinib mesylate. That's the same one that works in the Philadelphia chromosome positive chronic myelogenous leukemia. We found that from a tumor type called GIST. And this is how that fits into the standard and most common mutation. This is how it fits into the mutation we had. Here's the proline that's mutated in this patient, and the tail of imatinib sits right here. It doesn't fit. We looked at dasatinib, another drug that had been developed for totally unrelated reasons, developed to hit SARC, that toolkit of drugs that we have out there that are hitting many different things. If we can just figure out the right targets, we can move along. And its tail hangs out over here, way away from this mutation. It turns out from a number of studies in cell lines we developed that this is at least 10 times more active against this aberration. So again, we had taken this wonderful confluence, a drug toolkit, ability to characterize mutations, and found something important. As luck would have it, 3 weeks later, a patient comes in with melanoma. This patient had already been treated with imatinib, targeting KIT, even though KIT hadn't been sequenced, it had been done elsewhere, and had not responded. Had had every therapy known, 9 different courses of therapy, and we decided we would take a look and sequence all of what was going on in their tumor, and we found exactly this mutation. Now, we already knew they were resistant to this drug because they had received it, so we decided, and we got compassionate leave to treat with dasatinib. This is the patient. This is a PET scan. This is glucose uptake in the tumor that's in the pelvic cavity. This is all tumor. This is rectum here. This is all tumor. It's massive. This patient was in a wheelchair. This patient, we had to get special permission because they didn't have adequate performance status to go on clinical trials. This is what happened 3 weeks later. We shut this tumor down. Now, we didn't cure this patient. This was far too much disease. Patient got up on an oral drug, had 6 months of wonderful quality of life, felt better than they had for years. FDA won't even look at it. They don't care about quality of life. We can't prove that they had a longer life. Even if it was miserable, we can't prove that. However, this is a hit. It's a first step showing that the principle of looking at an N of 1 and moving forward can work. So I told you I'd tell you about our big project, Project Insanity. We are going to take the next 10,000 patients that come to the MD Anderson Cancer Center and characterize their tumor with 10,000 different genetic analyses. And we're going to direct them to different therapies based on this. We are going to ask, can we truly deliver on the promise of personalized medicine? At the MD Anderson Cancer Center, we're calling this a pilot project. Okay, so what are we going to do? All of our patients where we don't have a useful therapy today, where they failed previous therapies, or where they look like they're likely to fail therapy, we're going to do a CLIA, that's the regulatory process, compliant mutation detection, for any activating mutation in any gene that is ever shown to be abnormal in more than 5% of any cancer. We're also going to look at any case where there's a potential drug. All of that toolkit, we will look for every possible target. We're going to then do two things. The first one is do an N of 1 trial. Can we really deliver on personalized medicine? If Mrs. Jones has a mutation in her lung cancer, where we've shown that there's a drug that works in bowel cancer, we will test and see if she would respond. This is not how we do things. We treat lung cancer in one clinic, bowel cancer in another one. If she has a mutation in her breast cancer, and we've had a drug that has worked in renal cell carcinoma, we'll treat her with that drug. We'll never get statistical power to say each one of those drugs works, but rather we will ask if the process of simply saying this is the most likely drug to work for you will work. Will we be able to say that this type of process is going to benefit our patients? Our preliminary data already says yes. More importantly, we have 800 drugs that are targeting particular genetic aberrations. We need to match those drugs that are in trials to the right patients. Most of the time today, simply any patient who's available goes into that trial and we fail to see anything because we don't have the right mutation. And in other cases, some of these mutations are extremely rare. My favorite one, something I've worked on for over 20 years, a molecule called AKT, is mutated in 0.7% of cancers. We have 3 separate drugs that solely and beautifully target AKT. Think of doing a clinical trial. You're going to test 10,000 patients and find 70 that have the aberration. If you start as that as your goal, and it's $1,000 for each test, the company is going to laugh at you and it isn't going to happen. If instead we test for targets for those 500 drugs and amortize the cost across all of them, and it turns out that it's the same cost, it's actually less with this approach, to test for all 40 targets that we're looking at than it is for one. We can now start to fill those trials and rapidly determine who will benefit. So this really should make a difference. We may fail, but we will certainly fail big. Okay, here's the other challenge. This is what's happening with the cost of sequencing the human genome. 1990, $3 billion to look at 3.6 billion nucleotides. 2006, $20 million. Incredible. What an improvement. I'm not going to spend $20 million on 10,000 patients. 2010, it is now $500 to characterize the open reading frames of genes. It's still somewhere around $10,000 to look at the whole genome. But at $500, we can really start to do this. Now, that's not the charge. I don't want to have anything to do with what we charge people. This is what it will cost us. Can we afford not to do this? Can we afford not to ask if we can make a difference? By getting the patients on the right drugs, we're going to save money because we're not giving all of those drugs that don't work to patients who don't benefit. Can we really go to the concept of personalized therapy? We've established an Institute for Personalized Cancer Therapy at the MD Anderson Cancer Center. There will be an announcement next month as to who the head of that will be, and the goal will be to use this process to move the concept forward. I want to give you a case study, though. This is the problem. This is a patient who came in who had lung cancer, had all of the therapies that you could have in lung cancer and simply didn't respond. It was a relatively well-behaved disease. It was growing slowly for a while, didn't grow much for a while, and now it's starting to go quickly. We looked at all of the aberrations that we could look at using the T9 project, the 40 that we looked at, and found absolutely nothing. We went to our IRB and said, we've got these tools, let us sequence the genome of this patient and find out what's wrong. And the answer was, no, you may not do so, because you don't have a CLIA-approved test that would validate your results. Patient said, to heck with this, and went out and found somebody that would do it for them. This is what they found. And it turns out that this little aberration here is another one of those kinases that I've been telling you that we've got drugs that work on. We also know that this particular gene is mutated in other diseases and is seen to be mutated. So we said to our IRB, let us move ahead and treat this patient. We've got drugs that work. Sorry, you can't do so. You don't have a CLIA-approved assay. Please develop a CLIA-approved assay and come back to us. We pointed out that it would take 4 to 6 months to develop a CLIA-approved assay, and this patient is unlikely to live beyond 3 months. Can't do it. Patient went back, highly vested in his own disease, given this information from another site. I wasn't allowed to do so by our IRB. And started to ask around and said, does anybody have a CLIA-approved assay for TNK2? We couldn't find one. But then again, we weren't driven the same way. It turns out that the Moffitt Cancer Center has a CLIA-approved assay for TNK2. He found it. And so now we can go ahead and treat him. The question is where was our IRB? We have failed to keep up with this incredible change in technology by looking at compliance rules. We have a regulatory cause of failure today. Our regulatory processes have failed to keep up with research progress. We have to have a way to move forward quickly and efficiently so that we don't harm patients. That's their job. This patient's going to die in 3 to 4 weeks. So I'm not exactly sure how we can harm them by doing genetic testing. And we need to figure out how to move forward quickly with all of the regulatory groups, CLIA that manages tests, the FDA that says you have to have the statistics in a double-blinded test showing that patients don't benefit that have never benefited in before. We have to change the process. Now, it's one of the reasons I'm here. I can't change it. I'm a physician. I'm at a cancer center. They're not going to listen to me. The only people they're going to listen to are you. You're going to listen to people who say, not no, but hell no. We don't want this anymore. We're dying of cancer. We have major diseases. Yes, we want you to protect us from harm, but we need to balance that with benefit. And it's something that I hope you can do better than we can and move things forward. We have a regulatory traffic jam, and it is simply unacceptable. We're paralyzed. We can't move forward at just the time when we can make a difference. Now, this is a slide that worries me a little bit. I came to the MD Anderson Cancer Center, as you heard, in 1994. And in 1994, I had to put together a proposal as to what my department would look like. And this was one of the slides that I made. Now, it's actually not my version of it. I got it off of the web, attributed to 2 other people. But this is what we said. This is what's happening in our understanding of cancer. These are the new genome tools. These are the drug toolkit. And this is what's happening with patients. Incremental change that is far too slow. We have this incredible gap that we call translational research with an unbelievable confluence of opportunity to move forward, and we must deliver on that promise for our patients. Now, I've talked about the present and the future. Predictions are risky, especially when they're about the future. Every time we take a step forward, we see 20 different stoplights. This actually is a set of lights from Saskatchewan. This particular statement was originally attributed to Yogi Berra. It sounds like something he would say. If you go into the different sites, you'll also find that it's been attributed to Niels Bohr. It may make sense there. You can't even predict the present if you're a physicist. Turns out that baseball and physics have a lot in common. That ball moves all over the place for reasons I can't figure out. It turns out it wasn't from either. It was originally derived from something that Mark Twain said, a famous American humorist. And it's our goal to not fail and make sure that what I've told you about today is not humor, and we do move forward. Thank you.
For a few questions. So, is there a mic? We can get it around. If not, please shout and we'll try and repeat the question. So,
yeah, my question is, of the 10,000 people you expect in the trial, how many do you expect to have mutations that are addressable by the drug?
Yeah, okay, so you've asked one of the key questions, and it's a set of slides that I use in my more basic science presentations. We have been incredibly surprised by our pilot project. First pilot is just simply validating the process. We did 1,500 patients. And about 45% of those patients have mutations that we think are targetable. From what we knew in the past, or what we thought, we expected it to be 70 or 80%. We now understand why. It turns out that most of the targets in the drugs that we have are in relatively low grade, relatively well-behaved diseases that we cure by surgery and other approaches. The nasty diseases have a spectrum of mutations right now that we don't have good drugs for. That already is a major step forward because it's changed the way in which we design trials, it's changed the way in which we do a lot of things, but I would say it's a failure. It's not what we expected, and it's very bad news. Yes?
I have a question here. I have a mic, actually.
Could you share with us what countries you feel have a more enlightened compliance procedure?
Any? And I actually told a couple of my colleagues today that I may not be allowed back at MD Anderson tomorrow after some of the things I'm going to say. You know, I came from Canada. It's supposed to be United States, the free. I will tell you, this is the most regulated place I've ever been. The regulations for moving drugs forward are more complicated here than they are in Europe. They're more complicated than Canada. They're more complicated in any third world country. Right now, as far as I can tell, it is more difficult to move anything forward here than anywhere else. Indeed, many of the big drug companies now do their studies overseas. Now, that used to mean China, South America, where it was cheap. Now it's Europe because they have a reasonable regulatory environment.
I'd like a little bit of information about what you mean by targeting a mutation. Clearly, what you're targeting is a protein. And what are you trying to do? Make the protein not function? And how does that cause the cancer cell to die?
Okay, so in all of these cases, and I apologize for not being clear, we're not targeting the genetic aberration at all. We are always targeting the protein product. And almost all of the drugs that we have today are against two particular classes of molecules. Ones are G protein coupled receptors on the cell surface that perceive the environment, and we block the little piece of that that binds to a growth factor or a regulatory factor in the environment. We do the same thing with tyrosine kinase receptors. The vast majority of these drugs target an enzyme activity, and they block the enzyme. Turns out that these cells are addicted to this particular change. They're driven by the change in the protein that happens because of the mutation. And if we can target that selectively, we seem to be able to develop an adequate therapeutic index where the tumor cell stops growing or dies and the patient continues to live. We've spent a lot of time trying to figure out why that is. Our group, contemporaneous with Bernie Weinstein, who unfortunately passed away last year, and I now say it's his work, coined the term oncogene addiction. Cancer cells are misbehaving because of these genetic changes, and it turns out that then they are addicted to those genetic changes. They forget, and we can go through the whole systems biology approach of how and why they forget to use other molecules and other approaches. And they only have one way to grow and survive, and if we can find that and hit it, they have no choice but to die. Normal cells have many ways to get around that. And if we think about it, there are more cells in each of your bodies today than people have ever lived on the surface of the earth. There are more changes in those cells, mutations happen, and the cells are robust to those. Further, things in your diet and what you eat target any of the molecules once or twice. Your cells aren't addicted. They can relax, they can survive that event, but the tumor cells have no choice. They are truly addicted. You take away their addiction and they get very sick. And that's really the process that we've been working on. There are a few other reasons why drugs work, but that's the big one.
Other questions?
So I had a question regarding why do you think the FDA is reluctant to accept the strategy of like doing research on a specific subset of patients as opposed to like the large generalized study?
Okay, so one of the reasons for this is in the past, this is called a subset analysis. And there have been many subset analyses done where they really weren't done appropriately, where the subset was not very clear and it was a hope that maybe the drug would work. As we now are able to develop subsets that are robust, that we can define genetically and we know truly exist, we have to change. And change is a problem. I think you could do a much better job having a Colloquia or a Scientia on change because the whole world is resistant to change of any type. I've done it this way forever. Why would I change? Now, it turns out there are some reasons behind it that are a little more than that, but not much. We don't change easily. And basically, the FDA was charged with making sure that no harm was done. It came primarily out of thalidomide. Thalidomide is an absolutely incredible drug if you're not pregnant. So we didn't do a subset analysis. It is the best anti-nauseant we've yet found. It actually works beautifully against some tumors, but it's banned completely for nausea because it's a problem in a small subset of people, those that are pregnant. And so the goal of the FDA and the challenge that the FDA has is to make sure no harm happens. We've lost a term that is used called equipoise. That's the balance between harm and good. If I have a patient who is going to die in 3 to 4 weeks or 3 to 4 months from their disease, and I have a tumor like I showed you where no one survives at this stage, it should be the patient's right to make the decision of what they want to do. Now, it's a little hard to say what informed consent is because it's hard for me to be informed and I work in the area. But I don't think it's my business. I certainly don't think it's the business of an ethicist to tell this patient who has no other alternative and something might work, no, we just don't want to do it that way because there might be some harm. We've got to learn to balance benefit and harm. And right now, we have swung that pendulum over to where I can't get in trouble if nobody is hurt, and there is no penalty through the FDA if people aren't helped. And so it's a law that was put in place to prevent harm, and it's swung way too far, way too conservative.
Thank you. Well, I'd like to invite everyone out for reception afterwards. If you have additional questions, you may ask them, and I'd ask you to please thank our speaker for very enlightening and provocative.
This program is protected by a copyright and may not be redistributed in whole or in part without the express written consent of Rice Digital Media Services.